Okay, so part of a gene sequence from a eukaryotic cell will be displayed on the following slide. Note that the transcription will begin at the box GC base pair and will proceed from left to right. Transcribe and translate the sequence. So as mentioned on the previous slide, we're going to go ahead and start on G and C. It's boxed in on white there for you. And our coding strand, it's going to be on the top and template will be on the bottom. Now when it comes to transcribing, we have two choices. You can either use the coding strand. If you choose to do the coding, then all the letters will remain the same, except for every T, you're going to go ahead and switch that with a U. If you choose to use the template strand, then if you see a G, it's going to be transcribed to a C will be a G, a T will be an A, and an A will be a U. Okay, so now that you have transcribed either of those strands, it should look like the one in green. And that light blue box, it's going to be our promoter. So those letters that are included there will not be included in the final transcription. So now we're going to go ahead and look at either the coding strand or template, and we're going to look for our start codon. If you're looking at the coding strand, it's going to be ATG, and if you're looking at the pre-mRNA, it's going to be AUG. So we're going to go ahead and space out every 3 letters after that. It's going to help us throughout our translation process. And we're going to go ahead and refer to the chart on the bottom left there. So now we have our start codon. We're going to look for our stop codon. And it will either be UAA, UAG, or UGA. Now if we're looking at the coding strand, it's going to be the TAA. And if you're looking at the pre-mRNA, it'll be UAA. So now we have both of the start and stop codon. And if you continue to refer to your chart, as you can see, AUG will be translated onto MET. CCA is now PRO, and GAU is now ASP, and so on. And as I mentioned previously, the light blue line letters will not be included in the final transcription. Okay? So you should have that as your final translation and transcription.